Hello, everyone, and welcome to today's live broadcast, Comprehensive Genomic and Transcriptomic Analysis of Acral Melanoma, a Path Towards Therapeutic Development, presented by Dr. Winnie Liang, Assistant Professor, Integrated Cancer Genomics Research Institute, Collaborative Sequencing Center Director, Scientific Operations, TGen. We are excited to bring you this educational webcast presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. I'm Julie Simmeroff of LabRoots, and I'll be your moderator for today's event. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located in the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window. Or you can use the Q&A button to let us know that you're having a problem. This is an educational webinar and thus offers free continuing education credit. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your webpage and follow the process of obtaining your credit. Please join me in welcoming Dr. Winnie Liang. I will now turn the presentation over to her. In welcoming Dr. Winnie.
Hi, so today I'll be presenting you with a study that we performed and that was funded by the Melanoma Research Alliance as well as Stamp to Cancer. And so the title of my talk today is Comprehensive Genomic and Transcriptomic Analysis of Acral Melanoma, a Path Towards Therapeutic Development. So before I begin, I did want to highlight that this study was really built around a collaborative model. And so we had multiple institutions involved in the study, and the success of the study really relied on this collaboration. So I did want to highlight that we have clinical partners at Vanderbilt University as well as Memorial Sloan Kettering, and they provided really well annotated and clinically phenotyped samples for analysis. The samples were provided to TGen, where we performed next-generation sequencing, as well as subsequent genomic and transcriptomic analyses. Finally, as a result of our studies, there was orthogonal work that was able to basically be spearheaded from our original research, and that was performed at the University of Arizona and as well as Mayo Clinic in Arizona. And so again, while this is a data and bioinformatics topic, again, this wouldn't be possible without having high quality samples as well as a number of different collaborations. So just to give some background on acral melanoma, this is a rare subtype of melanoma. And so when we think of melanoma, we often think of the cutaneous form. However, acral melanoma occurs in only about 2 to 10, they comprise of only about 2 to 10% of all melanoma cases in the United States. It actually occurs more predominantly in non-Caucasians, but it is also the only form of melanoma that occurs at the same rate across multiple races. Compared to cutaneous melanoma, acral melanoma has a worse prognosis, as we'll see in the following slide. In terms of what we understand about links that may be associated with causing the disease, there is no obvious association with exposure to UV light, which is known to be a factor for susceptibility to cutaneous melanoma. In addition, there aren't any other family history or other environmental factors that have been known to be associated with this form of melanoma. In terms of what we know genetically and genomically about the disease, we have observed in some preliminary work that there is low frequency of BRAF V600 mutations in approximately 15% of acral melanomas. And there's as well as about 10 to 15% incidence of KIT mutations as well. And for the most part, the majority of cases are triple wild type, meaning that there is a wild type BRAF V600, NRAS, and also NF1 genes. Again, historically, most of the analysis that's previously been done in acral melanoma has been limited. It really wasn't until this year, until we performed our study, that we were able to get really do a full comprehensive analysis of acral melanoma. In addition to our study, there is also a publication published by Dr. Nick Hayward in Australia, who also performed whole genome sequencing, and that publication also came out this year as well. But overall, because of our limited knowledge of the disease, our goal now is to better understand the disease, both at the DNA and RNA levels, so that we can identify improved treatment options for patients. So looking at survival, as I previously mentioned, acral melanoma is associated with worse survival compared to cutaneous melanoma. You'll see in the left graph that the lower line, the lower dotted line, is reflective of acral melanoma, and survival is shown across the bottom, as well as the percent of patients who are surviving. And so for both of these graphs, both of the lower lines on both graphs are reflective of acral melanoma patients, and you'll see that compared to cutaneous melanoma, survival is much worse. And so again, this reemphasizes the need to better understand the disease so we can identify and develop improved therapeutics. In terms of what we know about mutation burden, there has been multiple studies that have looked across different melanoma subtypes. On the bar graph on the left, you can see that with regards to burden of coding mutations, acral melanoma falls somewhat in the middle compared to other subtypes, including cutaneous, mucosal, and uveal melanomas, with a mean of about 272 mutations. If we look at structural mutations by subtype, which is shown on the right-hand graph, again, you'll see that acral melanoma falls sort of in between both the mucosal and cutaneous subtypes, with a mean of about 80 structural variants. So just summarizing the workflow for this study, again, this was a collaboration among clinical sites as well as TGen and other collaborators as well. And so at Memorial Sloan Kettering and Vanderbilt, this is where patients were seen by the doctors on the study. They diagnosed the patients, performed a biopsy, and also did analysis of the biopsies. The samples were then forwarded over to TGen, where we performed extractions of DNA and RNA. We performed library preparations and then next-generation sequencing on the Illumina platform. From there, we initiated an analytical pipeline that was developed in-house and performed analysis across both the cohort and at the level of individual patients. So going back again to the clinical site that was involved in collecting all the samples, again, this was at Vanderbilt and Memorial Sloan Kettering. And what was really fantastic about this collaboration is that they maintain a really well-annotated clinical database. As a result, we have robust clinical and pathologic data associated with all the samples that we received and that were sequenced. So we were able to receive fresh frozen tumor specimens for each patient, as well as a matched normal. And so overall, we received samples from 16 patients from Vanderbilt University and 18 patients from Memorial Sloan Kettering. So overall, we had 38 total samples collected from across 34 patients. So looking at the breakdown of samples that we received about 15 primary tumors, 16 metastatic tumors, and we had a few patients where we received either multiple metastatic lesions or either a metastatic lesion and a primary lesion from the same patient. And again, we had 34 unique patients for which we have detailed clinical information as well as clinical follow-up. And this is just an overview of the patients that we had sequenced. Again, there was 34 total patients. But for 2 patients, we sequenced 2 tumors each, and for a third patient, as shown in green, we sequenced 3 total tumors. So again, we did have very detailed clinical information associated with each patient. And so at the time of the study, we input this information into an internal instance of cBioportal. And so I'll just walk through some of the different annotations that we have just to show the level of detail of information we have associated with all the patient samples. So we have information such as location of the primary, thickness of the primary tumor, as well as stage at presentation across all patients. Some other features that we have is the primary depth, if there's primary ulceration, and the number of lymph nodes that were impacted across all patients. And we'll see that, as you can see in the pie chart on the lower right, that 22 patients out of 34 had positive lymph nodes. Other characteristics that we captured include the race. See that the majority of the patients are Caucasian. In transit status, which is really a reflection as to whether or not the disease is spreading, as well as age at diagnosis. We also have metastasis status, as well as overall survival status for all patients as well. And so looking at the distribution of stages across the patients that we analyzed, the majority were stage 2 or 3, with only about 1 stage 1 and less than 5 patients who had stage 4 disease. So overall, the cohort of patients were advanced stage. Looking at survival of the patients in our cohort, the median overall survival was 86 months, and the median follow-up for the survivors were at about 36 months. And looking at overall progression-free survival, we had a median progression-free survival of approximately 18 months, with a follow-up for progression-free survivors at approximately 25 months. So looking at the experimental design that we implemented for the study, we used an approach, it's essentially using 3 different sequencing modalities to most comprehensively capture as much information as we can from both the DNA and RNA. In many studies, they've been moving towards using a targeted approach where you're not looking comprehensively at the whole exome or the whole genome. But we do think that limits us in our ability to do discovery work. And so we took a very comprehensive approach to our study. And so the first modality that we implemented was paired tumor normal deep exome sequencing using the Agilent V5 plus UTR baits. And this enables us to help capture low-level mutations in heterogeneous samples. Another approach that we also implemented was using long insert whole genome sequencing across paired tumor normal samples. And so this was an approach that we developed here at TGen. And what we do using this approach is essentially triple the size of the insert that you're sequencing. And in doing so, we triple our ability to identify a breakpoint in that region. As a result, this is a strategy for looking for structural events such as inversions, large deletions, and breakpoints, as well as this can be used to look at copy number changes as well. And so we don't sequence these genomes as deeply as the exomes. It's usually around the order of 5 to 10x for average sequence coverage, but the physical coverage is much higher because of the length of the inserts. And so it's typically in the order of 30 to 40x physical coverage. We also perform RNA sequencing on samples. And so this enables us to perform detection of RNA fusions. We can look for if there's any skewed allelic expression associated with any mutations we identified. We can also perform differential expression as well as pathway analyses. And we've also been using it to run our Neo-antigen analysis pipelines as well. So just to give an overview of the analytical pipeline, our sequencers they talk directly to our LIMS system in the lab. And once sequencing is complete, we kick off BCL conversion to generate FASTQ files, and these are fed directly into our DNA and RNA pipeline, which all fall within a single pipeline. Following an analysis of both DNA and RNA, we generate emerged VCF, which we reinsert into a separate server where we annotate all the variants with things like the gene name, any descriptions or any prediction algorithms, as well as population frequencies. So looking more closely at the DNA pipeline, there's 2 major components to the pipeline. The first shown here is relatively probably well known and established across other institutions that also do sequencing analysis. We perform alignment using BWA. We perform marking duplicates and sorting using Picard. We also do joint indel realignment and also recalibration using GATK. And so the resulting BAM file is used for variant detection. And so we have 3 different primary analyses that are done in this pathway. The first is looking for point mutations and indels, the small indels. And so we use 3 separate tools. These include Mutex, Seurat, and Strelka. Because we use an ensemble approach, we do require that an event is called by at least 2 of these 3 tools to reach a list of variants of interest. We also have internally developed tools here at TGen for looking at copy number and also structural variant detection. So here's a high-level view of the RNA pipeline. It's not too dissimilar from the DNA pipeline. We use STAR for alignment of data. We also use Picard for marking duplicates and sorting, as well as GATK for microalignment and recalibration. The resulting BAM is used for further analyses, and so we use TopHat for detection of RNA fusions. We use CuffDip and DSeq2 for looking at differential expression, and we also use GATK and SAM tools for looking at allele frequencies of mutations. So looking at the metrics that were generated, we sequenced just over 16 high-output V3 flow cells on the Illumina platform. And this encompassed over 47 billion total reads. And so in sum, we had generated over 3.8 terabytes of data. And so the data output and the number of reads generated for each batch of analysis is shown at the bottom. And you'll see that our Q30s for each of those different sequencing modalities are all greater than 86%. So one of the first analyses that we can pull or perform after doing sequencing is looking at tumor cellularity, and this can be estimated using allele frequencies after we perform SNB detection. And you'll see that for the most part, the tumor cellularities across the samples are relatively high, probably averaging probably around 30 to 50%. There are a few samples with lower cellularities, and this helps us to better interpret the data once we take a closer look at the variants that were called. So this is another analysis that it falls formally outside of our pipeline, but is another analysis that is really beneficial, particularly in the context of melanoma. After performing SNB detection, we can go in and look at what the actual base change is, what base changes are actually occurring at each of those variants. And so using the Watson and Crick pairing, the different possibilities of base changes are shown in this figure. And you'll see that the majority of the changes are occurring from C to T. And so it was about 55% of all SNBs that we detected had a C to T transition. And this is particularly relevant, again, for melanoma, because in cutaneous melanoma, it is well known that there is a UV signature associated with the disease. And what this means is that over 60% of C to T transitions are occurring in dipyrimidines. And so we decided to look at that in our cohort of acral melanoma. Although we don't expect to see a UV signature, we thought this would be relevant to check. And so when we did a cohort level analysis of C to T transitions, only about 39% of those events were occurring in dipyrimidines to show that there is no UV signature present at the cohort level. However, we also went into each individual subject and also performed this analysis. And what was interesting is that we saw that 2 patients actually showed a UV signature in their tumor, and the 3rd patient was trending towards having a UV signature with about 59% of their C to T transitions occurring in dipyrimidines. And so this is a bit of a busy slide, but it summarizes a lot of our work from doing the SNB detection, as well as our copy number analysis and structural variant analysis. So along the bottom, you'll see the patient numbers associated with the different tumors. And across the top, there are, on the 2 bar graphs on top, those show mutation burdens with respect to coding point mutations for SNBs or copy number changes as reflected as CNVs. And we also broke these down following the TCA subtypes that's typically used to characterize cutaneous melanoma. And so we could look in more closely at this, and we'll see that there are 18% of the patients that we sequenced showed some level of BRAF alteration. With respect to NRAS or KRAS mutations, 15% showed alterations, and about 9% showed alterations in NF1. Overall, from our entire analysis, we had identified point mutations in we identified about 9,500 total point mutations and 72 small indels. However, looking at only coding events, we identified a median of 42 point mutations. And what was interesting that we observed from this analysis was that there was 4 patients who received treatments prior to us sequencing their samples, and so they demonstrated higher levels of mutation burdens. And so their median coding SNVs were approximately 75 as opposed to 42 when we look at the entire cohort. We also used InteGen and MutSig to identify putative drivers in our data set. And so when we did a cohort level analysis, we found that NRAS and BRAF were significant putative drivers in this data set. And what was interesting is that in the patients that had NRAS events, 3 of them have a well-known activating Q61K mutation that has been reported in cutaneous melanoma. Again, looking at the KIT mutations, there was actually only 1 patient who had a KIT alteration. And this was actually a little bit unexpected because in previous reports, it was reported that 11% of ALMs have a KIT alteration. And there's also multiple ways you can look at the data set. So we did look at changes that might be associated specifically with metastatic status or whether the tumor was primary or metastasis. We did do a specific primary-only analysis as well as a MET-only analysis. Following the analysis, we found that BRAF was still a significant putative driver in both cohorts if we did this division. However, what was interesting is that the mutation burden was higher in the MET samples as opposed to the primary samples. So looking at copy number changes from our entire analysis of the entire data set, we identified about 1,100 somatic CNVs. Following this analysis, we performed consensus analysis of the CNVs. So we wanted to see what was the most recurrently occurring changes across the entire cohort. And this consensus analysis was performed using GISTIC-2. So following GISTIC-2 analysis, we identified 48 total CNVs, of which 40 were deletions and 8 were gains. And these are summarized here on this slide. And you'll see that some interesting events that we identified include gains in PAC1 and also CDK4, as well as deletions in CDKN2A, PTEN, and also NF1. We also did a division of the sample set into primaries only and METs only. And we observed that in only primaries, the PTEN deletion retained significance. And in the MET only sample group, there's MDM2 gains that were significant to that group. So moving on to the structural variant analysis, overall we identified roughly about 2,500 different somatic breakpoints across 31 patients. And of the 31 patients, 74% demonstrated some form of a structural variant. And so in this graph, we summarize a number of structural variants that were identified across all the patients. And you'll see that patient 14 had 683 somatic breakpoints identified, and they all fell on chromosome 12 to show evidence of chromothripsis. For the most part, the range of value of structural variants was more heavily around 100 to 200. And so this is in line with previous reports that there is a heavier structural variant burden in acral melanoma. So we also looked at the most frequently impacted chromosomes by the somatic breakpoints that were identified, and we found that in at least patients with a wild-type BRAF, the most number of structural variants were found to impact chromosome 5. And on chromosome 5, there's 2 genes that were heavily affected, and these include ADCY2 and CLPT-M1L. And what was interesting is that, as you'll see in later slides, we did find increased levels of changes in TERT, and CLPT-M1L lies upstream of TERT. So another analysis that we also ran was also assembling RNA reads to see if we can find supporting RNA data for these breakpoints that we identified. And looking at the ADCY2 or the CLPT-M1L SVs, we did find that there was a presence of supporting RNA fusions in 4 of the 6 patients that showed events in these genes. And so again, this sort of reemphasizes the benefit of having both DNA and RNA level data. And so here I just summarize all the copy number changes and also structural variants that were detected across the entire cohort. You'll see that in the center, these are the yellow lines reflect translocations, and you'll see that it is heavily impacting primarily chromosome 5 on the lower right-hand corner. So we also performed a sort of a pathway type style analysis using the DNA level data, and so pulling out key recurrent pathways that are commonly reported in melanoma, we looked in these pathways to see if the genes were mutated or impacted in any way in our cohort. And so on the left-hand side, looking at the survival and proliferation pathways, we found that 66% of genes in this, 66% of patients demonstrate some sort of alteration in the survival or proliferation pathways. And you'll see for the most part, a lot of events are associated with a gain, with the exception of P10 and NF1, which are demonstrating loss of function. We also observed TERT alterations in 1/3 of the patients. We also observed changes in cell cycle progression, with 51% of patients showing alterations in that pathway. And with respect to apoptosis and senescence, we observed changes in 17% of patients. So another type of analysis that can be performed using this type of data set is looking at therapeutically actionable somatic events. And so we did this by going through each patient's data and seeing if there's associations with any drugs that are currently available. And what was sort of encouraging was that about over 70% of patients demonstrated therapeutically actionable events. And some of them are listed here. So for example, with the BRAF V600E or V600K event, possible therapies that can be considered include dabrafenib, vemurafenib, and trametinib. And this is relevant for 4 patients. The CDKN2A focal deletion, which was prevalent across a number of patients, can be targeted using palbociclib, ribociclib, or CDK4/6 inhibitors. And so again, this is just another demonstration of the utility of having this type of data set and how it can be used to sort of infer the benefit of sort of precision medicine type work. So again, the results that we found support the etiology of micromelanoma and also identify potentially novel therapeutic options. And again, because we have such a large data set, there are a number of different analyses a number of different additional analyses that can be performed. And so another approach we took to the RNA-seq data was that after pulling out differentially expressed genes, we looked at the topmost differentially expressed genes. So these were about 420 genes, and performed unsupervised clustering of these samples and genes. And so the heat map shown here is the results of this clustering analysis. So you'll see along the bottom, these are the patients. So each patient corresponds to a different column. The rows that go horizontally correspond to specific genes. And what was interesting is that when we clustered these genes, we found that there was 5 clusters that broke out. And you'll see these as the red, yellow, green, blue, and purple clusters. And these clusters are associated with specific processes. And so you'll see that the red cluster is associated with protein translation, ribosomal processes, and also nonsense-mediated decay. And you can go through and see the rest of different pathways associated with these clusters. And what was also interesting is that we did see that certain pathways help to differentiate samples based on whether they were primary or met. And so you'll see that sort of large chunk down at the bottom of the heat map where you see that the majority of the metastasis samples are green, and the majority of the primary samples are red. And so this is really in line with actually what has already been reported in cutaneous melanoma, specifically that keratin genes in METs are downregulated, whereas they are upregulated in primaries. And we're similarly seeing that here in this April cohort. Another analysis that we also performed integrating both RNA and DNA data is Neo-antigen analysis. And so this analysis it really lets you see the level to which there are potential new peptides that may be expressed as a result of a coding somatic alteration that was identified through exome sequencing. And so we looked at neoantigen expression across all the patients, as well as HLA expression with the MHC class 1A, B, or C molecules. And so this is definitely of interest since there's been a sort of a wave of use of checkpoint inhibitors. And so for our cohort, there was 22 of 34 patients who received checkpoint inhibitors. In cutaneous melanoma, response to checkpoint inhibitors has been described, but this hasn't been described yet in micromelanoma. And so similar to what we see in cutaneous melanoma, we did see a correlation between neoantigen burden as well as mutation burden. We also we did a sort of a preliminary analysis looking at the correlation between mutation neoantigen burden and a response to checkpoint blockade. Unfortunately, we don't have enough patients to power that type of analysis, so we weren't able to derive a conclusion from that. However, there were 3 patients in our cohort who demonstrated a complete response to either anti-PD-1 or anti-CTLA-4. And interesting, they had both lower mutation and also neoantigen burdens. Another interesting result that we found in our study was the identification of events impacting PAC1. And so we observed copy number gains as well as overexpression of PAC1 in about 12% of patients. And these patients were mutually exclusive of those that showed BRAF, RAS, or NF1 alterations. And this has, again, PAC1 has not actually been specifically implicated in acral melanoma. And so this is why we thought this was interesting to lift up, since it has been defined as a potential therapeutic in cutaneous melanoma. Following our analysis of both the DNA and RNA data, we also found that there was a number of different alterations impacting TERT. And so in cutaneous melanoma, it is well known that there is a percentage of patients who demonstrate TERT hotspot mutations in the promoter region, which leads to activation of TERT. However, this hasn't been specifically reported in acral melanoma, although one study did find that 9% of acrals did demonstrate a TERT hotspot mutation in the promoter. And so in our analysis, we found that aside from these TERT promoter mutations that were also similarly identified in 9% of our patients, there were a range of different types of events also impacting TERT. And so we have them summarized here. So we see copy number gains impacting TERT. There's a number of different breakpoints, 1-point mutation. And we also identified that in 2 patients for whom didn't have any somatic TERT alterations, each of them had their own rare germline SNP in TERT. And these events were predicted by a prediction tool, FATHOM, which stands for the Functional Analysis Through Hidden Markov Models. It was predicted to be damaging. And something also to highlight here is that we did observe TERT expression across all samples. The graph probably doesn't do justice to it, but we do see some level of expression of TERT across all samples. And under normal conditions, we wouldn't expect to see this. And so this suggests the possibility of TERT activation in many of the samples. And so overall, we saw that 41% of the patients demonstrated some level of TERT alteration in their tumor. Given this finding, we worked with Mayo Clinic in Scottsdale, Arizona, to perform some downstream functional assays to look at the efficacy of inhibiting TERT in acral melanoma. And so we used 3 different cell lines. The first, SMC09, had a TERT CNV gain in that cell line. There was another cell line, SU2C 001002, that had a TERT promoter mutation. And we also used a normal melanocyte line as a control. And so both of the 2 acral melanoma lines that we looked at are derived from actual patients. And so what we did was we used telomerase inhibitor 9, which is a commercially available inhibitor, and treated each of these cell lines for 72 hours. And we found that using 2.5 micromoles, we did the treatment of using the inhibitor resulted in a decrease of over 75% viability in the Acral cell lines, whereas it did not highly impact the normal melanocyte cell line. We also looked at the expression of TERT mRNA in these cells following treatment with the telomerase inhibitor, and found that there was no expression and no change in the normal melanocyte line, and there's decreased expression of TERT mRNA in the 2 Acral cell lines that are treated with the telomerase inhibitor. And so the details of all our analyses are described in our publication in Genome Research, which we published earlier this year. And so another aspect of our analysis was that we really understood the value of being able to share our data with our collaborators and also just with the scientific community. When the study was ongoing, we did have an internal instance of cBioPortal, and we also had servers that we can use to easily transfer data between collaborators. And after completion of the study, we have dumped the data into dbGaP so it could be available by so it's available for analysis by other investigators. We've also placed our data on cBioPortal. So if you go onto the public website for cBioPortal right now, you have access to our data, and it's under the melanoma heading. So I did want to briefly touch upon some of the orthogonal studies that we worked on as a result, and sort of really as a result of the work we did through our primary work. So based off of our findings that TERT inhibition may be an effective way to kill acral melanoma cells, we did work with Dr. Lawrence Hurley at the University of Arizona. And he does really interesting work where he's able to develop inhibitors that specifically target a motif that occurs in the human genome, and this is the G quadruplex or G4 structure. And so this structure is specifically enriched in promoters of oncogenes and also in the beginning of the first intron of genes, as well as in telomeres. So he was able to design an inhibitor that was found to successfully lock down the TERT promoter inhibitor, and in doing so, it can repress TERT expression. However, removal of this particular inhibitor can revert the expression of TERT back to overexpression. And so it was able to regulate TERT expression, and we thought that this is a particularly meaningful approach to potentially regulate TERT expression in cells that are TERT dependent. In addition to the G4 quadruplex analyses, we also have a collaboration again with Mayo Clinic, and they were developing PDX from the samples that from the samples that were collected from the patients we sequenced. And so they initiated a number of different models, and right now they have currently 10 models that are ready for testing. And what's really beneficial about this is that it allows us to do drug treatments and further functional analyses, particularly in patients that may have more of a quiet genome. For example, one of the patients did not demonstrate significant number of structural variants or even point mutations. However, they did have an NRAS mutation. And so we can really focally sort of do analysis in this type of context using this PDX model. So in terms of current and future directions, we do have a study that was just recently initiated, and this is headed up by Dr. Will Hendricks at TGen and Dr. Lance Cowie at Baylor Scott and White. And the goal of the study is to continue to characterize the landscape of TERT alterations in mucosal and also acral melanomas. And secondary, the goal is also to look at the effect of changing or TERT alterations on the function of telomerase. And we also want to look at the impact of TERT inhibition in both mucosal and acral melanoma. And so again, I want to reemphasize the importance of having a collaborative model. Given again that this is a data and bioinformatics track, the success value and really the utility of bioinformatics really hinges on getting high quality samples, having clinical annotations, and doing comprehensive analyses. And so it's the combination of both clinical resources as well as bioinformatics and genomics that really is going to drive our ability to make a translational impact on patients. And so I'd like to acknowledge a really large team of scientists who were involved in this study. The study was headed up by Dr. Jeffrey Trent here at TGen, and as well as our clinical collaborators, Dr. Jeffrey Sossman and Charlotte Arian at Vanderbilt and Memorial Sloan Kettering. Alex Sekulich at the Mayo Clinic, along with Jessica, really spearheaded the TERT cell line analyses, as well as developing the PDX models. Lawrence Hurley, he was our key collaborator for identifying an approach to potentially regulating TERT expression. And Ji-Yun Lee from the Samsung Medical Center provided the Acral melanoma cell lines that we use for analysis. And again, this effort culminates as a result of all the work from a number of different informaticians and scientists as well. And I'd be happy to take any questions.
Thank you, Dr. Liang, for that informative presentation. It's time for Q&A. If you have a question you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click on the send button. We'll answer as many of your questions as we have time for. If we are unable to get to your question, we will follow up with you via email after the presentation. Let's get started. Our first question is, were you able to use the different exome, long insert, and RNA data sets to validate mutations? An RNA data set to validate mutations?
Yes, great question. And so that really speaks into the benefits of having multiple sequencing modalities from the same samples. And so we were able to validate some of the events. Specifically, if we look at coding events, 12% were validated. 12% of the coding SNPs identified through exome sequencing was validated through long insert whole genome sequencing. I'm noting the fact that we did have low sequence coverage, lower average sequence coverage of the long insert whole genome data. However, looking at those same coding events, 90% were validated by the RNA-seq data. And so again, 90% of the coding events we identified were expressed.
Great, thank you. We'll wrap up with one more question, and that question is, aside from the PAC1 and TERT findings, were there any additional results that were striking?
So yes, in addition to the TERT and PAC1 findings, there was quite a bit interesting things that we found. One of and as I mentioned during my presentation, was the identification of a USB signature in 2 of the patients, which was very unexpected. After reviewing the data with the clinicians, we did sort of realize that the possibility that I'm needing a better biomarker for disease, because it is possible that because acral melanomas most often occur in plantar regions or sun-protected regions, there may be misdiagnoses where they may actually reflect a different type of melanoma or a cutaneous melanoma. And so that was something that was educational for both the scientists and clinicians, and it helps us to better understand how we can better improve sort of diagnosis for patients.
I would like to once again thank Dr. Liang for her presentation. Do you have any final comments?
No, again, I just wanted to thank everyone for attending. And again, I've mentioned this multiple times during my talk, but again, the utility and the benefit of doing bioinformatics analysis really requires the use of high-quality samples. And so I hope to encourage people to continue to collaborate and work together.
Thank you once again, Dr. Liang. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through January 12, 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.